Hello students, welcome back to our biology class. Today I'll be starting off with the chapter number 2, Reproduction in Flowering Plants. In chapter number 1, we have discussed reproduction in general. Now, let us see how the reproduction occurs in flowering plants. You already have an idea about this chapter. You have read about the preliminaries of this chapter in class 10. So I'm going to start off the concepts from class 10. Remember the structure of a flower? What did we study in class 10? That flower is the reproductive structure in plants. This flower contains what is known as the sporophylls, the structures which produce the spores. That is the gametes. Now, recall that a typical flower consists of 4 whorls, namely calyx, corolla, androecium, and gynoecium. Calyx and corolla are the outer 2 whorls which do not participate in reproduction directly. So, they are called the accessory whorls. But androecium, which is the collection of stamens, and gynoecium, which is the collection of carpels, are the male and female reproductive structure. So, this chapter we will divide into 3 segments. The first segment of this chapter will comprise of the pre-fertilization events. Then the second part is fertilization events, and the third part is post-fertilization events. In pre-fertilization events, we mainly have the formation of pollen grains, formation of embryo sac, and pollination. In this module, we will see the formation of pollen grains. So, all of you know that where are these pollen grains or the embryo sac found? The pollen grains are formed by the structure called stamen, and collection of stamen is androecium. A typical stamen consists of a stalk-like structure called filament and a bulb-like structure at the top of which it is called the anther. This anther produces pollen grains. Similarly, the female reproductive structure, which is the collection of carpels, that is gynoecium, this gynoecium has one or many carpel depending upon the type of flower. Each carpel again has 3 well distinguished parts. The first one is the stigma, which is the landing platform for the pollen grains, which are released from the anther. They land on the feathery, sticky area called stigma present at the top of the carpel. Then there is a long stalk-like structure which is called style, and at the base of which we have a bulb-like structure resting on the thalamus, which is called ovary. This ovary can contain one or multiple ovule. And within the ovule, the embryo sac is formed, which we'll see in the second part of the pre-fertilization events. But first, let us see the development of pollen grains in the anther. Now, to know how the pollen grains are formed in anther, we need to learn about the structure of an anther. If we cut transversely the anther what we will find? That each anther is divided into two parts or lobes. These lobes are connected at the back with the help of a thick tissue called connective, and in the frontal side it has a deep groove. These two lobes are called the theca. So an anther is basically a bitheca structure or ditheca structure connected by connective at the back and a deep groove on the frontal side. Now, if you see through each lobe is again divided into two cylindrical structure, or it harbors two cylindrical structure, which are called the pollen sac or microsporangia. Thus, each theca has two sporangia. So, overall 4 microsporangia are present. So, we call an anther to be tetrasporangiate. Dithecous tetrasporangiate structure. These microsporangia later matures into what is known as the pollen sac. And within this pollen sac, the pollen grains are produced. Now the question is, how is this anther produced in the flower? See student, of the decision to form a flower is taken way earlier than we find the blooming of actual flower. At the top of the stem, what do we find? A vegetative bud. A vegetative bud which forms the leaves, the further stems. This vegetative bud, under the influence of various hormones like florigen, indolacetic acid, gets converted into a floral bud or floral primordia. This basically is nothing but differentiation of the primary meristematic tissues which are found in these primordials. Then this meristematic tissue gets differentiated into a group of tissue which is known as archisporial tissue or archisporial cells. These archisporial cells then further give rise to two types of cells, the outer parietal cells and the inner primary sporogenous tissue. The outer parietal cells gives rise to the 4 walls of the anther. The outer parietal cells give rise to the 4 walls of the anther, and inner sporogenous tissue, they form the pollen mother cells from where the pollen grains are produced. So what are these 4 walls of the anther? The outermost wall of the anther is called epidermis, and this is a continuous wall which encompasses all the 4 pollen sacs. Then we have a layer of flat cells called the endothecium. Inner to it, we have 3 to 5 layers of middle layer. 3 to 5 cellular layer of middle layers. And the last inner or the innermost wall comprises of what is known as tapetum. Now this tapetum individually surrounds the 4 microsporangia. The formation of the microspores, the pollen grains from the pollen mother cells, which act as the microsporocyte or the meiocytes, which undergo meiosis, is known as microsporogenesis. So, the process of formation of microspores from the pollen mother cells is known as microsporogenesis. A pollen mother cell is a diploid structure. It has two sets of chromosomes. This pollen mother cell, also known as microsporocyte, undergoes meiosis. And as you all know, meiosis produces 4 haploid cells. Now, initially these 4 haploid cells are joined to each other with a callose layer. Now, this callose layer later on gets disintegrated with the help of the enzyme secreted from tapetum, which is called callase. And each microspore released from the microspore tetrad develops into a pollen grain. So this conversion from microspore to a mature pollen grain is nothing but differentiation. There is no further division but differentiation of the cell. So what differentiation occurs? The pollen grain gets covered, or it remains covered with 2 layers of wall known as sporoderm. The outer layer is known as the exine and the inner layer is known as the intine. The outer layer is composed of one of the most toughest organic material called sporopollenin. This sporopollenin is an oxidative polymer of carotenoids, and this is resistant to heat your acid, alkali, and there is no enzyme known which can digest this sporopollenin. The layer of exine is not continuous. There are few regions where this sporopollenin is absent. Those regions are known as derm pore or germ pore. These germ pores are the areas from where the pollen tube generates. The inner layer is a continuous one and is formed of pectocellulose. Inside this, there are actually a larger vegetative cell which has irregular shaped nucleus, and there are plenty of fructose, unsaturated fatty acids, amino acids present in the cytoplasm of this vegetative cell. Within the cytoplasm of the vegetative cell, there is another cell which floats in the cytoplasm, and this is known as the generative cell. And this has a tapered nucleus. Now, in 60% of the angiosperm, a pollen grain is released in this two-celled stage, where there is a generative cell, the larger generative cell or tube cell, and the larger vegetative cell or tube cell and the smaller generative cell. But in rest 40%, the generative cell again divides into 2 nuclei known as the male nuclei, and this represents the 3-celled stage of the pollen grain. Now, these pollen grains, upon pollination gets deposited onto the stigma of the carpel. These pollen grains are variedly used. They are used as nutrient supplements because, as I said, they are storehouse of your unsaturated fatty acids and fructose, very nutritious, and you'll often find them in the racks of supermarkets. Pollen grains also are stored in very low temperature in liquid nitrogen, which is known as cryopreservation, so that they can be used for artificial hybridization in the form of pollen bags. These pollen grains also cause large number of allergic reactions in the body. Itching, reddening of the eyes, sneezing, skin eruptions, etc., which is known as pollen allergy. The carrot grass or Parthenium, which was imported with the wheat by the British, is one of the major allergen in the North Indian region. So, this is how a microspore is produced in the pollen sac. Once a mature pollen grain is formed, the endothecium layer dehydrates and it disrupts. It bursts out due to dehydration, and this liberates the pollen grain into the air outside, which are then carried to the stigma by various agents. Now, these three layers of the anther, that is epidermis, endothecium, and the middle layers, actually serve protective function. They do not have any other function. They are mainly protective in nature. But the tapetum layer, that is a very important layer, and that is actually nutritive in function. It provides nutrition to the growing microspore as well as the pollen mother cells. At the same time, they secrete sporopollenin, Calais enzyme, indole acetic acid, etc. These tapetal cells are identified due to their dense cytoplasm and binucleate structure. Means these cells have binucleate structure. The cytoplasm is deeply stained, so it is dense, and the ploidy level is multiple. That is, the nucleus is polyploid. And this is how we identify the tapetal cells. So, this is about microsporogenesis. In our next module, we will discuss the formation of embryo sac. Thank you.